Organoleptic characters of coriander fruit. The coriander fruits are usually brownish yellow in color, and they are having aromatic taste because of the volatile oil, and they are having spicy and characteristic taste. Okay, that's why we are using this coriander fruits in a spices, that is in the food. And its extra features, it is also a cream of carb. Okay. These are also psilospermous fruits. Okay. Next, before starting this microscopy of powder drug and microscopy of the crude drug, we have to know what type of microchemical test we have to perform to identify the microscopical characters. Okay, this microchemical test mainly done for the powder drug and also for the crude drug. The first one is we have to take the crude drug powder or section, then we have to add chloroglucinol plus HCl solution, which is in 1:1 ratio, so that all the lignified sclerenchyma and vascular bundles which are present in the crude drug or powder will turn to pink color. Okay? In the same way, when the crude drug powder or the section is treated with alcoholic citric acid solution, all the environ grains which are present in the powder and also the crude drug will turn to yellow color. Okay, next, Sudan red berry solution is for oil globules. It is mainly for fixed oil globules and also for the cuticle. Okay. So when this powder or crude drug is stained with the Sudan red berry solution, all the oil globules and cuticle will turn to red color. Next, microscopical characteristics of the powder drug. Powder drug, when you observe this powder drug under microscope, it is usually, we can see these four characters specially. Okay, these four characters of powder drug helpful for the identification of the crude drug. Okay, one is sclerenchymatous layer, next endocarp and endosperm and vitae. Okay, these three are same as a phenyl, but it is somewhat different and it is a crucial for the identification of the coriander fruits. Say sclerenchymatous layer. Next, okay, first one is the sclerenchymatous layer. This sclerenchymatous layer actually present in the mesocarp of the fruit. Here in powder we can see a group of fusiform fibers running wavy. Okay, they are usually fusiform means the ends are tapered. Okay, fibers are having their ends tapered at one end. So fibers are having tapering ends. Okay, and these fusiform fibers are usually wavy and they're crossing each other. Okay, in this picture you can see these fusiform fibers usually crossing each other, and these are lignified sclerenchymatous layer. Okay, sclerenchyma we already studied, it is a lignified tissue. So here also the sclerenchymatous fibers are lignified. And next one is endocarp. Same as funnel, here also endocarp is having paraquatry arrangement. That means group of parallel cells are arranged in different directions. Here, these also having thin-walled dignified cells with polygonal cells. Okay. Here, endocarp is having paraquatry arrangement of dignified cells, and these polygonal shaped cells are actually mesocarp. It is not an endocarp. Mesocarp present on the endocarp present on the mesocarp, so it will come to cell. Okay. We already observed in the funnel also. Next part is endosperm. It is also same as funnel. Okay, there is no difference. Polygonal shaped parenchyma type of cells. These are cellulosic parenchyma cells with aileron grains, oil globules, and micro rosettes of calcium oxalate crystals. Okay. And vitae. Here also same vitae is a few vitae. We can see few vitae fragments. Okay. These are usually yellowish brown in color, and they are also made up of thin walled small parenchyma type of cells. Okay, these parenchyma type of cells are formed together to form a bunch like structure. Okay. Vitae, endocarp, endosperm, these are similar to fennel, but the difference is here sclerenchymatous layer. Okay. For example, if I have given two powders in a lab, one is fennel powder, other one is a coriander powder. By observing its organoleptic characters like color, odor, taste, you can easily identify the drug. Okay, even though you are unable to identify which is coriander powder, which is fennel powder, then you have to go for the microscopical study of the powder. Okay, that for that you have to take the powder and slide, and you have to add some reagents like chloroglycerol HCl. You have to observe them under microscope, and you have to check the powder for the presence of this sclerenchymatous layer. Okay, if sclerenchymatous layer present, it is a coriander fruit. If reticulate parenchyma, if you observed any reticulate parenchyma in the powder, it is a fennel fruit powder. Okay, this is only the difference. We can easily identify these two crude drugs by their microscopical characters. Here you can see this is a endocarp, okay, par-quad rearrangement of the endocarp in effect in a coriander fruit. Here you can see a group of parallel cells are arranged in different directions on mesocarp. It is the sclerenchymatous layer. I already told you it is a lignified fusiform fibers which crossing each other. Okay, some are going like this and some are in this direction. Sclerenchymatous layer looks like this under microscope. It is a lignified, so it will stain pink color with chloroglycerol. Let's see. Next, microscopical characters of the crude drug. Okay, not a powder. This is a crude drug. This is the transfer section of coriander fruit. Okay, when you take the entire transfer section from the entire fruit, it will have two mericarps. This is one mericarp part and the remaining is another mericarp part. Okay, but usually while drawing the detailed diagram, we usually draw only one mericarp. Okay, we also studied only one mericarp structure because it is a similar to other mericarp. Two mericarps are similar. Okay, they are similar in their microscopic. Okay. It is actually having two parts, two mericarps. It's an entire fruit. Schematic diagram of the cremocarp. And I already told you any fruit will have two parts specially. One is a pericarp, other one is a seed. Okay, pericarp is again divided into three parts. One is the epicarp, mesocarp, and endocarp. And below the endocarp, there will be a seed. Okay, seed. The outermost layer is a testa, and below the testa there will be endosperm. In endosperm, we can find an embryo, right? Okay. So the outermost layer is here epicarp. I already told you. In leaf, the outer layer is called epidermis. In fruits, the outer layer is called epicarp. And the middle layer is here mesocarp. From here to here, okay, from this that is below, just below from the epicarp to above from the above for above the endocarp. It is a mesocarp. It will have sclerenchyma strip in the middle. Okay, this dark colored band is here sclerenchyma strip or sclerenchyma layer. Okay. And here it is a double line, right? The outer line is actually indicating endocarp and the inner line is a seed coat. And this part is the endosperm and this empty space is raphe. Raphe is nothing but longitudinal band on the seed. Okay, and this part is here caprophore. I already told you it connects two mericarps. It is actually made up of sclerenchyma type of tissue. It connects two mericarps, and these are vitae, two circles here, and these three on dorsal surface are lacunae. Okay, what is lacunae, what is vitae, I will explain in the next slide. Here it is a dorsal side, and this is the commissural or ventral surface of the fruit. Okay. microscopical diagram of camel foot. And it will be pericarp. It is having two parts, pericarp and a seed. Okay. Pericarp is again having three parts, epicarp, mesocarp and endocarp. Okay. Epicarp here is made up of only single lined cells. Okay. You have to draw it only within a single line of polygonal to rectangular shaped tabular cells. Okay. And the epicarp is covered within a thin layer of cuticle, which is made up of suberin. Okay, that's why when we stain that section or crude powder with Sudan red clay solution, this cuticle also turns to red color. Okay, and one important character is here. These epicarp cells are having calcium oxalate crystals. Okay, some cells of the epicarp are having calcium oxalate crystals. Okay. And below this epicarp, from this from below to epicarp, it is called mesocarp till this. Okay. From here to here, it is a mesocarp. So the middle layer is called mesocarp, and here this middle layer is again divided into two layers: outer layer and the middle layer. Sorry, three layers: outer layer, middle layer, and inner layer. Okay. Just the area above this, the area above this sclerenchyma strip is called outer layer, and this sclerenchyma strip is called here middle layer, and below this sclerenchyma strip it is called inner layer. Okay? The inner layer is made up of parenchyma type of cells which are tangentially arranged. Okay, here you can see the size of this inner layer parenchyma cells is larger. Okay, here it is made up of larger reticulate, larger lignified parenchyma type of cells which are tangentially elongated, okay, are tangentially arranged. But here also it is a parenchyma, that is outer layer also parenchyma, but it is not lignified. Okay, we already studied in the tissue side, parenchyma is actually not lignified, and the cell walls of the parenchyma are usually made up of cellulose. But in case of coriander, inner layer of the mesocarp, okay, it is also made up of parenchyma, but the cell walls are made up of lignin. So it is a lignified parenchyma. It is also one important character of the coriander fruit, which is helpful for the identification of this fruits from the other fruits, or which is helpful for the differentiation of coriander fruits from the another fruits. Okay, this middle layer is here sclerenchyma layer or sclerenchyma strip. Okay. Uh, First, we will discuss about this outer layer. The outer layer is made up of parenchyma type of cells which are tangentially arranged. Okay. And here you can see this side is actually dorsal surface, right? And this one is a commensural surface or ventral surface. On dorsal surface, you can see lacunae in the outer layer of the mesocarp. This part is called lacunae. Okay, here you can see four lacunae, right? One, two, this one is three, and four. Four lacunae you can see on dorsal surface, right? And lacunae is nothing but the empty space. Okay, it is a gap in the mesocarp. It is not like vitae, which is having which is a oil tube. Okay, it is not an oil tube. It's just an empty space or gap in the outer layer of the mesocarp. Okay, outer layer is this is only the outer layer. Only it is made up of parenchyma type of cells, and it is also having lacunae. Okay, and, before discussing this middle layer, uh, I will explain this remaining part, okay, because it is very important. I have to differentiate each and everything here. Okay. Before discussing that, we have to know about endocarp. Here also same. Endocarp is made up of um, paraquat, that is parallel cells which are arranged in different directions. Okay. It is also having paraquatry arrangement, same as funnel. This entire part. Okay, here. This one is actually endocarp layer. Below the endocarp, you can see one layer, one line cells, which is here, which is called here testa. Okay, testa is made up of single lined yellowish brown colored cells, right? And testa is nothing but here seed coat, and this part is raphe. Raphe is nothing but one longitudinal ridge on the seed. Okay, this endosperm is nothing but seed only, right? Seed part is having one longitudinal ridge on their surface. That is nothing but raphe. This part is raphe, and this part is the endosperm. Endosperm here, same as spindle. It is also made up of polygonal to hexagonal shaped cells. Uh, it is also having oil globules, fixed aileron grains, and calcium oxalate crystals. Okay. And next about this commissural surface. Okay. Here you can see two vitae on commissural surface. Vitae is nothing but oil tube. Okay. Two vitae on the commissural surface, but no vitae on the dorsal surface. In coriander fruit, vitae is not present on dorsal surface. Only two vitae are present on uh, commissural surface or ventral surface. Okay. Instead of vitae, here we can see lacunae, but the function of lacunae is not same as vitae. Next one is here caprophore. We already studied that caprophore is made up of two strands of sclerenchyma type of tissue. It connects this mericarp with another mericarp of the fruit. Okay. Simple. Okay. One important thing here is middle layer of the mesocarp. This middle layer of mesocarp is made up of uh, two type of sclerenchyma type of tissue. Okay, it is actually sclerenchyma strip or sclerenchyma layer, but here it is again having two type of sclerenchyma tissues. Okay, one is tangentially elongated sclerenchyma strip, other one is longitudinally elongated sclerenchyma strip. Okay, uh, this part is here tangentially elongated sclerenchyma sheath. Okay, this one. And here in gap, that is in between the sclerenchyma sheath, here it is, this one is a longitudinally elongated sclerenchyma. Okay, on longitudinally elongated sclerenchyma we can observe five vascular bundles. Okay, five circles here you can see, these are nothing but vascular bundles. Okay, on every longitudinally arranged sclerenchyma we can see five vascular bundles. What? Here, 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 and here. Right? These all are lignified. So when you stain this sclerenchyma sheet, when you stain this transfer section of coriander, this entire part will turns to pink color. That is sclerenchyma layer and the inner layer of mesocarp, because it is also made up of lignified parenchyma cells. Okay. This is about microscopy of coriander fruit. Mm, yes. For color differentiation, I usually draw the diagram by using different chalks, different color chalks. But sometimes for the differentiation, I will use another chalk also. Okay, because here the sclerenchyma is actually lignified. So I have to draw with the pink color. But for the differentiation from this longitudinal sclerenchyma, this is a tangentially elongated sclerenchyma, and this part is here longitudinally elongated sclerenchyma. This part. Okay. These all are lignified, but for the differentiation only, I draw it with drawn it with blue color. Okay, but it is also lignified. And here you can see five vascular bundles on the surface of longitudinally elongated sclerenchyma. Okay, and four lacunae on the dorsal surface. This is the inner layer of the. It is also I have to actually draw it in a pink color because it is also lignified parentage. It's endocarp, endosperm, raphe, okay, caprophore, epicarp. Okay. Here I drawn this testa with an yellow color. It is usually yellowish brown in color, right? So how to draw this diagram I will show you later. We already discussed all these characters, right? Epicarp, mesocarp, endocarp, and then seed. Okay. It is actually very blurred diagram. Okay, for the color differentiation only, take this one in the slide. Here you can see this is a green one, here is a epicar, and these are lacunae. Okay, here somewhat dark pink color is showing, right? It is nothing but vascular bundles, and below this vascular bundle it is a longitudinally elongated sclerenchyma, and this is a tangentially elongated sclerenchyma, and this part is reticulate parenchyma, and endocarp, endosperm, two vitae, raphe, and caprophore. So it is very blur. Then substitute. Before drawing this, we have to know what are the substitute adulterants of this coriander fruit. Okay, there is no adulterant for this fruit. Adulterant means only the exhausted coriander fruits, okay, which are having an inferior, which are inferior quality drugs. But here it is a substitute. Bombay coriander is a substitute. Bombay coriander fruits are having less volatile oil when compared to the original one, that is Coriandrum sativa. Okay. They are somewhat bigger in size, and their shape is also different. They are in somewhat ellipsoidal in shape, but actually our coriander is having subglobular in shape. Okay? But these are ellipsoidal in shape and bigger in size, and they are having less volatile oil. These are Bombay coriander. It is a substitute, and he uses. Okay? Coriander fruits are used as a carminative because of the volatile oil. Okay, that means they expels the gas from the stomach. The aromatic, right, because of the volatile oil, they are used as aromatic, flavoring agent, CNS stimulant, and spice. The most common use is spice. Right? In everyday uses dhania powder in every dish, every spicy dishes, right? So the major uses as a spice. But coming to the other medicinal uses, this coriander fruits and seeds are used in the treatment of various urinary tract disorders, urinary tract infections. Okay, they are good for the kidney. Okay. This coriander leaves, coriander fruits, the entire plant of this coriander is helpful for the kidney health. Okay, before that, I already told you that how to draw this diagram. Um, First, I will stop sharing this. Simply, I will show that now. Okay, share whiteboard. Share. However, it is possible to put this mouse. First, draw. Does not possible for me with a mouse. First, simply you draw first the structure, the shape of this, and then you draw some epicarp layer. Okay? And I told you that tangentially elongated cells, right? Tangentially elongated cells means somewhat like this. ఒక straight గా కాకుండా ఏదో ఒక సైడ్కి you just bend the cells. Okay. When the cells are arranged like in a tangentially elongated way, tangential elongated cells in వచ్చిన ప్రతిసారి you draw like this. And what I told you, sclerenchyma straight, right? Tangentially elongated sclerenchyma is first you draw like this. ఒక baby structure draw చేసేయండి around this inner layer of the inner layer of the mesocarp. Then you fill this space with tangentially elongated cells like this. With the mouse. Okay, while taking the class, I will explain one more time by using mobile phone. 9 minutes. Next, about longitudinally uh, elongated sclerenchyma. You have to fill this gap with the longitudinally elongated sclerenchyma. Here I am using another color for just differentiation, but it is also lignified, so it will also pink. Just draw one circle. Okay, circle and dot, circle and dot. Okay. Don't leave any space in between this. Circle and one dot, circle and one dot. It is when you draw this one longitudinal elongated sclerenchyma, it usually like bunch of looks like bunch of grapes. Okay. And above this, just draw some small sized circles and then one dot inside. Okay. Again, I told you that above this longitudinally elongated sclerenchyma, there are five vascular bundles, right? So you have to draw five vascular bundles. You have to draw vascular bundles like this: circle, circle, circle. Last one. In the same way here also longitudinal sclerenchyma and then again five vascular bundles. Longitudinal sclerenchyma and again vascular bundles. And I told you that inner layer of the mesocarp is having some larger lignified parenchyma cells. The size మాత్రమే they just increase the size of the cells. And then shall we cells. And you know how to draw this endocarp, how to draw endosperm cells. Okay. And lacume. Lacume is not like Witte, it's just like this. Okay. It's just a gap or empty space in the outer layer of the mesocarp. But in case of Witte, you have to draw some globular shape and then you have to draw some parenchyma cells around this Witte. Right? Next is about caprophores. Okay, here usually we have some caprophore. First you draw one circle, then you draw some small circles inside and place one, put one dot. Okay, put one dot inside the circle, circle and dot, circle and dot. It is also look like bunch of grapes. This is the way of drawing the coriander transfer section. Here the very simple is this both coriander and fennel fruits are in your syllabus. Okay, with this fruit, both are umbelliferous fruits, both are cremocarp, both can be divided into mericarps. Right. The only difference is first morphology. Okay. Morphologically, the fennel fruits are oblong in shape, but these coriander fruits are subglobular in shape. Okay. And fennel fruits are also having ridges on their surface. Coriander fruits also having ridges on their surface. But coriander fruits are having both type of ridges, primary and secondary ridges. Primary ridges are wavy and they are invisible. They are not clearly visible. But in case of the secondary ridges are straight, they are prominent. Okay, we can when we touch the surface, we can easily sense that secondary ridges. Okay, and here the stylopad is in fennel, the stylopad is bifurcated. It means it is having two calyx teeth. But in case of coriander, it is a five calyx tooth. It has five calyx tooth on the stylopad. With five calyx in it. And next, coming to the organoleptic characters, both are having aromatic odor and both are having characteristic and spicy taste. Okay, but fennel is having sweet taste and sweet odor. Okay, coriander do not have any sweet taste or sweet odor. And one more important thing, fennel fruits are greenish or yellowish brown in color, but these coriander fruits are brownish yellow in color. Okay. and both are chromocards, both can be divided into mericards, and the two mericards are connected by the caprophore. And next, coming to the powder characteristics. Reticulate parenchyma is one important diagnostic character of the fennel fruits. But uh, in coriander fruits, we cannot identify any reticulate type of parenchyma. We can see lignified parenchyma, and the important character is here sclerenchymatous layer. Okay? You can see sclerenchyma type of fibers in the powder and also in the root drug. It is one important thing. And one more important thing, witte. Okay, in case of fennel fruit, we can see six witte, four on dorsal surface and two on the commissural surface. But in case of coriander, only two witte are present, and they are present at the ventral surface. Okay, on dorsal surface we cannot see any witte. We can see only lacunae, which are just an empty space. Okay, and here mesocarp is having three layers. Outer, middle, and the inner layer. Inner layer is made up of lignified parenchyma, and the cells are somewhat larger, right? And here, sclerenchyma again two types, longitudinally elongated and tangentially elongated. Okay. On longitudinally elongated sclerenchyma, we can see five vascular bundles, right? This is only the difference between fennel and coriander microscopical characters. Uses both are used as a carbonative. Okay? Both are used as a carbonative, both are aromatic, both are when compared to the fennel, coriander is a stimulant. This is about coriander fruit, and remaining drugs we will discuss in the later classes.